Okay. In this chapter, chapter 15, which is called Host Defenses 2, specifically focuses on the third line of defense. And when we talk about third line of defense, we're basically talking about two main players, the T cell and the B cell, and what they do. They do amazing things. But the first thing that we're going to talk about is the characteristics of the third line of defense. It is different than the first line and the second line of defense. Remember, I told you that first line and second line of defense is innate and non-specific. They know how to do things one way. If that helps you, they're happy. If not, go look somewhere else. The look somewhere else might be this. This, you might decide, okay, if you don't want to help me, I'm going to call in the big guys here. This is basically because the third line of defense is what is called specific. It actually decides on how to kill something one way, but how it kills something else will be completely a different way. So it produces a specific line of attack depending on which pathogen it's fighting. So it can produce a line of attack to kill E. coli a certain way. And like I said before, salmonella a different way. But how it kills E. coli will not be how it kills salmonella and vice versa. And in a sense, it is said to be specific. So the approach is specific to the pathogen that it's fighting. That's what it means. So the third line of defense has specificity. It has something else very interesting, memory. It remembers the bad guys that it was challenged with in the past. And we take advantage of that when we do vaccination. And you'll see that. So the first time it sees a pathogen, you'll be sick. Because it's still trying to figure out ways of, okay, how do I deal with this guy? And while it's trying to figure those things out and developing the immune system, the antibodies, and you're sick during that period of time. But then as soon as it figures out how to do it, the next time it sees it, assuming you survived the first one, it says, aha, I've seen you before. And I remember what you were like, because I have memory. And this time, you're not going to hurt me like you did before. It's amazing. So we can take this to our advantage through vaccination. When we inject a component of the pathogen in you, not the pathogen, because the pathogen would get you sick. No. We want to use the system to our advantage. We want to prime your immune system so that if you see the real thing, you'll already be ready. You will have already figured out a way to kill it. And we call that vaccination. We inject you with a component of the pathogen to stimulate the first response. To stimulate, more specifically, the memory. So that the next time you see it, assuming it's now going to be the real thing, you will not get sick. And we call that vaccination, and you'll see that. But we wouldn't have been able to do that if your third line of defense mechanism did not have memory. If they did not have memory, then we couldn't do that. If we inject you with a pathogen that has been inactivated, it will do nothing because you will not remember the event. So, because we learned that the third line of defense develops what's called memory cells, someone thought of how do we take advantage of that, and that's what we call vaccination. And you'll see that later towards the end of this chapter. Antigenicity. The antigenicity is the ability of the antigen, the foreign protein, to stimulate your immune system. See, not every antigen can stimulate your immune system, or not every antigen can stimulate your immune system to its potential. if an antigen can really stimulate your immune system, it is said to be highly antigenic. And these are usually these guys. The larger the antigen is, the more antigenic it is. Because it has more epitopes to choose from. And I'll explain to you what that is. Hang in there. So the larger the antigen is, the more antigenic it is. Now here's another thing. If the antigen is protein or complex things, like made up of complex things, it's better. Protein is better in antigenicity. Now if you combine both things, a large protein, oh, that's the best thing. Large and the fact that it's protein would make the best antigens, meaning it's the one that would stimulate your immune system to its potential, to its maximum or maximal potential. So that's the combination, that's the trick here. Large in size, in molecular weight, and protein. Those are more antigenic. That's why we can make antibodies out of degraded or denatured exotoxins, because those are proteins. And we cannot make antibodies out of the endotoxins, because the endotoxins are what? LPS. The LPS, the polysaccharide. They're not proteins. So, if we look at a protein, let's say this is a protein, and the boxes that I'm drawing here are basically stretches of amino acid sequences. So whatever, let's say glycine, arginine, arginine, glycine, whatever. And let's say histidine, proline, whatever. So these are referred to as epitopes. That's an epitope, that's an epitope, that's an epitope, and so forth. Epitopes determined by basically amino acid sequences. So of course, a protein with this size will have, let's say, 5 epitopes. But you can tell a protein that is larger would have what? More or less epitopes? More. See, that's why the larger proteins are better than the smaller proteins, because there are more epitopes to choose from. So, what are we choosing for? What are we doing here? Well, your immune system will recognize that these epitopes are foreign, because these are, for example, bacterial proteins. They don't belong to you. But it won't be able to necessarily identify all of them as foreign. If it did, that would be really good. If your immune system says, yeah, that's foreign, that's foreign, that's foreign, that's foreign, that's foreign, then even the small one will be antigenic. Because you'll have a lot of epitopes that your immune system, your antibodies can react to. That would be awesome. But it's not like that. This epitope is a sequence of amino acids. You may have the same sequence somewhere in your proteins. So it says, no, I saw that in my host cells. I can't tell if that's a host cell or a protein or not from that epitope alone. You don't want it to attack that one, because that means it will attack yours, your host cells. You want it to attack the epitope that's in bacteria, but not in anywhere in your body, in your cells. So let's say it's this one. Let's say it's in this one. Let's say this one. Let's say this one. Let's say this one. So the ones that I have purple are bacterial. I mean, they're all bacterial. But these, you may also have. You don't want an antibody to form against this one, because even though it will attack the bacterial protein, it will also attack your tissue. You want your antibodies to attack these because they're bacterial epitopes. And then you don't have them. So your antibody is going to target the bacteria, not you. But which one do you think will be more antigenic? The one that has more to choose from. This is the reason why this one is better as an antigen, because it doesn't just have one to choose from, it now has 4 to choose from. So this one is more antigenic than this one. This one will stimulate your immune system much better than this one will. And it all has to do with the availability of choices of epitopes to choose from. The interesting thing is this. If I take this one out, look. If I take this out, just on its own, just by itself, with nothing else attached to it, this would cause the immune system to be stimulated. This one would not, because it's too short. You see that? Even though it's antigenic in this major, big, huge protein, it's a component of this big protein, making the whole protein antigenic. On its own, it doesn't do anything. So it's not just a factor of it being bacterial, it also has to do with the size of the protein that it's attached to. When you take one of these out like this, that's actually called a hapten now. Haptens on their own do not stimulate the immune system. Even though as part of a larger protein, they would. But if you take this one by itself and inject it, just this part here, inject it into a mouse, nothing will happen. So it still needs the molecular weight component. So the problem in research is that we may not be able to isolate this protein all the time. If I can isolate this protein and inject it in you to get your immune system stimulated, okay, I'll do it. Then I don't have anything to worry about. But what if this protein cannot be isolated for whatever reason? And I know that this is antigenic, or this one, but too bad, I can't get the protein out. So we do this. Again, the human mind is unbelievable. When we're up against a problem, we don't just give up. We decide to either synthesize this part, and there's machines for that. We can actually synthesize this part. So we get this. If we inject this by itself, we will not get an immune response. But we can do this one. We can take this hapten and attach it to something called a carrier molecule to mimic the large molecular weight. Now that one will stimulate your immune system. So a hapten on its own does nothing. Attach it to a carrier molecule, inject it into the mouse, then you'll get an immune response. So we're tricking the system. I know I can't isolate this protein for whatever reason, but I know that epitope is antigenic. Well, synthesize the epitope, attach it to a carrier, and inject it. You'll get an immune response. Of course, none of this stuff would work unless we did research on these things. So through research, we discovered that. Alloantigens, these are just words now for this slide. Alloantigens are antigens that basically molecules of one person injected by mistake into another person that does not like those. For example, a mismatch in blood. If I give you blood sample that does not belong in you, that would be considered an allergen. You're going to have a massive attack, and it can actually kill, anaphylactic shock there, if not picked up on time there. So mistransfusion can cause a huge problem. All right, so mismatches of tissue would be considered an alloantigen as well. So when we do tissue transplantation, we look at the donor samples, MHCs, along with a whole bunch of other cell markers, and try to match it with the recipient, with the patient. You might not get a perfect match, but you'll come as close as possible. And then rely on possibly immune suppressants. So a lot of people that get donor or transplantation organs, they live the rest of their life on immune suppressants to prevent the immune system from attacking the new organ. There are some bacterial toxins that are referred to as super antigens because they stimulate your T cells at 100 times their normal stimulation level. So they're not just antigens, they're super antigens, to the point where it can actually lead you into toxic shock syndrome, and that can be fatal. And it all has to do with the release of chemicals, massive release of chemicals all at one time, trying to ward off the infection. And your body is overcome by that response. Okay, so we switched gears a little bit. The major histocompatibility complexes, we talked about one. There's actually two more. So it's a total of three: MHC1, MHC2, and MHC3. We actually talked about MHC3, but we didn't call them MHC3, we called them the complement system. Are we catching that part? Remember there's 30 proteins that are circulating in your blood, we call them the complement system? That's actually MHC3. That's this one. They're soluble. They're in your blood. They're not attached to your cells. If they do attach to a cell and put a hole in it, hopefully it's a bacterial cell, not your cells, because that would be really bad for you. So, look at the difference between the three. MHC3 are soluble proteins in your blood. They don't attach to the surface of your cells. They're circulating in your blood. And when they do attach to the surface of a cell, hopefully it's a bacterial cell, and it kills it. Your MHC1 is different. It is attached to the surface of your cells, of all of your cells. It marks you for who you are, except for the circulating red blood cells, because see, it attaches to all nucleated human cells, and the only cell that is not nucleated in your body is your circulating red blood cell. And there's a reason behind that. Physiology hopefully will teach you that. I'm not saying that the stem cell that produced the red blood cell does not have a nucleus. No, that one has a nucleus. But mammalian red blood cells do not have a nucleus, the ones that are circulating in your blood. And there's a reason for it, a couple of reasons. So all your other cells have MHC1 and they mark you for who you are. Remember, your immune system is going to check this out. Yeah, that's Dr. Saad, leave him alone. Thank you. So I don't have autoimmune disease, right? The time it doesn't recognize that, then it starts attacking my host cells, then I will have autoimmune disease. And sometimes that happens. It forgets the lesson that it learned. Now, there are other types of white blood cells. They're called macrophages, dendritic cells, and B cells. You need to know that. They have another MHC called MHC class 2, MHC2. Well, obviously, they also have MHC1. So they will also have MHC1 because all nucleated cells have MHC1, and these guys have a nucleus. But they also have MHC2. And we'll see what the function of MHC2 in these three cells are. Very important function. I'll give you a clue to what the function is right now. It's how they present antigen to the T cell. So T cells are very good at what they do. But the problem with T cells is this. For you to activate them, you need to tell them who the antigen is. And then once you activate them, now they know what to do. So I'm a T cell. I'm sitting here not doing anything because I can't, because you haven't shown me who the bad guy is yet. Once you do this, watch, a macrophage, a dendritic cell, or a B cell will do this. Attach to the pathogen, take a piece of the pathogen, and send it to the surface on the MHC2 saying, hey, that's the bad guy. Now watch, it was actually part of the pathogen. So what happens is, the macrophage, for example, will phagocytose the pathogen. The PAMP, the PRR, the phagosome, the lysosome, the phagolysosome. Do you remember all that? But while it's doing that, look what it's gonna do. It's gonna take a piece of it, send it to the surface, and attach it to the MHC2. This is the MHC2. And that's the antigen, because it's part of the bacteria, see? And it's saying to the T cell, hey T cell, whoever has this is the bad guy, because that's where I got it from. This is the bad guy. Do something about it. That's how you activate T cells. T cells must be presented with the antigen for them to be activated. So we say that these three are involved in antigen presentation to the T cell. How? Through their MHC2s. Every time you present an antigen, if you're a macrophage, dendritic cell, or B cell, to the T cell, it's going to be on the MHC2. Unless you are virally infected. If you are virally infected, you're presenting the antigen on MHC1. Do you remember in the last chapter that cell that was virally infected that sent an interferon out to warn the neighboring cells? Do you remember that one? And I told you it's going to take a piece of the virus and put it on the surface of the cell saying, hey, I know I'm infected. I know what you're about to do to me. Just get it over with. What I'm saying is a virally infected cell will present that viral particle on MHC1. Why MHC1? Because all your cells have the ability or the possibility of being virally infected, and all your cells have MHC1. Do you see that? So if my liver cell is infected with a virus, it's going to present the antigen on MHC1. Why? Because it has MHC1. If my lung cell has a virus, it will present the viral antigen on MHC1. Why? Because it has MHC1. It's amazing how this works. Any cell in your body can potentially be infected by a virus. Specific viruses, of course. But once they're infected, how are they going to let the T cells and the natural killer cells know that they're infected? They're going to take a piece of the virus and present it on MHC1. So the same thing with this one. If this cell got virally infected, they will present on MHC1. But things like bacterial antigen or whatever, it's going to be more on MHC2 to stimulate the T cells. All right. So the surface of the B cell is a busy place. Besides the fact that it has MHC1 to mark it for who it is, to present viral antigens on it. Besides the fact that it has MHC2 for antigen presentation to the T cell, it also has what is called a B cell receptor, which is an antibody. A B cell receptor, the only difference between a B cell receptor and your standard antibody circulating in your blood is the fact that the antibody circulating in blood is not attached to anything. It's soluble. It's mixed in your plasma. But the B cell receptor is the same exact antibody, but it's anchored on the surface of the B cell. That's the only difference between them, really. So an antibody is technically a circulating B cell receptor, whereas a B cell receptor is attached to the surface of the cell, of the B cell. So there are a whole bunch of receptors on the surface of all the cells. Here are some examples of that. The antibody is a Y-shaped structure, or even the B cell receptor, because it's the same thing. It's a Y-shaped structure made up of a heavy chain and a light chain. It also has what's called a constant region. The brown region here is the same, it's called constant, but this region here is variable, meaning when you look at two different antibodies, the C is the same, but the variable region will be different. That's why one antibody can recognize the red antigen, but another antibody recognizes the blue antigen. Antibodies are very specific to which antigen they bind to. Let's say this antibody binds the blue one. It will bind the blue one here and the blue one here. This variable region and this variable region, also called the antigen binding site, is now going to be specific to the blue one. And whatever it's specific to here, it'll be specific to here. It's blue and blue. You're not gonna have blue and then it's not gonna be specific to one antigen and a different antigen. No, it gets what's called the same shuffle, and we'll talk about that. You have another antibody that recognizes the red one. They're very specific. That's why the third line of defense has memory is one thing, but in this case, specificity. Remember that. It will program a certain way of killing. It will kill E. coli a certain way. It will kill Salmonella a different way. What I was implying here is it will produce an antibody against E. coli and a different antibody against Salmonella. That's what I was implying there. The constant regions, the heavy chains these are actually 4 different peptides, but the 2 heavy ones are attached to each other through disulfide linkage. So we're talking about cysteines in this region. And this one is held by disulfide bridges and disulfide bridges here. This region here is called the FAB region. So this arm of the Y and this arm of the Y is called the antigen binding fragment, the FAB region. More specifically, the variable region is where the antigen binds. This region here is called the FC region, the crystallizable fragment. And it's involved in other things, and we'll see if we can touch up on that later. Now, how antigens fit in the antigen binding site is kind of mimics the substrate and the enzymes to some extent. It's all based on the shape of the antigen and the shape of the hypervariable region of the FAB region, the variable region of the antibody there. If it has a perfect fit, you will get a very strong activation. If it has no fit, no activation. That's obvious. But you can also get a weak activation when it fits somewhat. So you can get a whole wide range of activations there, from very strong to no activation. And it has to do with how the antigen fits in the antigen binding site. Kind of mimics substrates and enzymes in that sense. This is another one. This is a real cool one. How is the variable region produced? And I kind of give you a hint of that. It's all about the shuffle. Think of, what's that game at the casino there? Blackjack. I don't know if you've ever played blackjack, but it's the dealer comes with a whole deck of cards, right? Like 8, 9 of them, because they want to make sure they win, not you. And they shuffle them. Imagine the dealer giving you part of that shuffle. Hey, that's you. And then they take the deck again and they shuffle it. And they give me a shuffle. So you have a shuffle and I have a shuffle from the same deck of cards. What's the chance that her shuffle is going to be the same exactly like mine? Very little. Considering that they usually bring out like 9 decks of cards. It's not just one deck. It's the same thing. The variable region is produced through something called gene rearrangement. This is not meiosis, it's actually through mitosis, which is a weird thing here. You're getting gene rearrangement during cell division for this gene. So in some cases, this shuffle is produced. In other cases, this shuffle is produced. This one. So this antibody has this shuffle. If you notice, the same shuffle on this arm is the same shuffle on this arm. And if this shuffle recognizes the blue one, then it's going to recognize the blue one here and the blue one there. Because the same antibody will have the same 2 variable regions. But this antibody, because of this shuffle, will only be specific to the blue one. That's it. What if you want one for the red one? That's a different shuffle. That's a different antibody. Here's the amazing thing. The amazing thing is that you are born with all the possible shuffles. And this one it just blew my mind the first time I learned it. You are born with all the possible shuffles that you can ever need. I'm not saying you will have to use all of them, no. I'm saying you have them in case you come in contact with the blue one. I have an antibody that recognizes this one if I come in contact with it. Well, what if I never do? I still have the antibody, it just never gets activated, you see? I still have the cells that can produce this antibody, they just never get activated. Thank God I never came in contact with it. But if I do, I have the cell. So you are born with all the different shuffles possible. And it's just a matter of exposing you to that particular antigen to activate that particular cell that has that shuffle. And we'll see how that plays a role later here. Do you remember the FC part of the antibody that I talked to you about? This part right here. That's involved in complement binding. I told you in the previous chapter that the pathogen, the bacteria moving by the complement 1, C1, is tagged with an antibody. So it looks like this, watch. The Y-shaped structure is attached to the bacteria and the FC part is sticking out. The C1 protein grabs onto the FC part. So that the FC part is used in complement, specifically C1, because the antibody is the bridge between the C1 and the pathogen, the classical pathway. So the complement 1 will bind to the FC region of the antibody, while the variable region is binding to the pathogen. That's why I called the antibody is the bridge between them. So that's one of the functions of the FC component. It has other functions as well. Enhanced phagocytosis. The FC part plays a role in opsonization. See, a white blood cell can phagocytose bacteria. So for example, it might go grab onto the bacteria. It might be a little slippery, you see? But eventually it'll say, okay, I got it. The next time it goes to grab onto it, it says, man, I wish it had some handles or something so I can grab onto it better and just get rid of it. That's the antibody. You attach an antibody to it, that's your handle, the FC. So the FC component of the antibody helps in enhancing phagocytosis, because when the phagocyte goes to grab onto the bacteria, it has got something to grab onto, and it just grabs it this time. So that's how it enhances phagocytosis. But then the phagocyte will be interacting more with the FC, grab onto the FC, drag in the bacteria. These antibodies come in varieties here. The IgG, IgA, that's a dimer, this is a monomer, this is a pentamer. The constant region can come in different lengths. The IgE is involved in allergies, allergic reactions. So some of your mast cells and the basophils will have these on the surface. And if the variable region happens to be a shuffle that recognizes pollen, by chance, then you're going to be allergic to pollen. So we all have mast cells that have these on their surface, but if they get activated, then you produce the inflammatory response. So it's all about the variable region and what they happen to react with. So if you're allergic to pollen, then you have an IgE with a variable region that recognizes pollen, that activates the mast cell to release histamine. And the allergic reactions are the result of the histamine release. Your body is reacting to the histamine release. IgM is a pentamer, IgA is a dimer, and this is the J region that links these things together. The J region, the joining region, is the one that helps make things dimerize and pentamerize. When you first contract something, you will always be producing IgM first. Your first line of attack is an IgM antibody. But your long-term immunity is IgG. So sometimes the doctor will order an antibody test against something. And he or she will ask, is it IgD or IgG? Sorry, is it IgM or IgG? You have an antibody, but which type? IgM or IgG? Because it tells the doctor, this is a recent infection. You've had this for a while. If they come back and say, yeah, he has an antibody for it. Which kind is it? IgG. Oh, he's had it for a while. That's the idea behind that kind of test. The IgAs are typically released in secretions like breast milk, for example. They always tell you should breastfeed your child. It protects them. Because in breast milk, there are some antibodies secreted and given to the child who is at this time immune technically compromised to some extent because the immune system has not fully developed yet. So you're helping the child ward off the various infections and diseases by breastfeeding. The IgA antibody type will be in breast milk and other secretions. IgD is the Bcell receptor. So if we go back here, I said the Bcell receptor is an antibody. I'm going to add the Bcell receptor is a surface receptor antibody of the IgD type. That's just the thing that's going to add here. And then it can get secreted once the B cell is activated. What about the T cell? No different. The T cell is a cell and it has receptors too. It has a T cell receptor that also through gene shuffling, gene rearrangements, produces a whole variety of different T cells. So you have a B cell that can recognize this, and a T cell that can recognize this through the shuffling of genes. So the T cell receptor may not look like the B cell receptor, but the antigen binding site of a T cell receptor is also produced through gene rearrangement. But it has other receptors called the cluster of differentiation, CD. You may have heard of these receptors before, and you did when we talked about HIV. We said HIV attacks the CD4 cells, right? T cells that have CD4 cells. So you have T cells that have CD4, you have T cells that have CD8, a whole bunch of CDs, hundreds of them. Some T cells, a lot of T cells actually have, or T cells may have one type of CD that is in common with all of them. So I cannot identify which T cell is which using that one, because they all have it. So let's say, for example, I'm just giving you an example, CD3. Let's say they all have CD3. Then I can't tell you if this is a T helper cell or a cytotoxic T cell or a memory, whatever, by looking at CD3. Because if they all have it, then how am I going to know that? If we all wear blue jeans, then I can't distinguish people based on blue jeans, you see? If we're all wearing lab coats, then I can't tell who's who by simply looking at the lab coat. We look at these CDs, the cluster of differentiations, that some cells have, but all the other ones don't. For example, the T helper cell has a CD4, but the other ones don't. The cytotoxic T cell has a CD8, but the other ones don't. So we can use which type of CD the cell is producing to identify which T cell it is. It's very useful when we're doing blood tests and stuff like that. Bear with me, please. I need to just finish this. The development of the B cells and the T cells, they originate in the bone marrow, like we talked about before. They are the lymphoid lineage. They originate in the bone marrow. So both the B cells and the T cells originate in the bone marrow, but where they mature is different. The B cells mature in the bone marrow. So they both develop in the bone marrow, but the B cells stays at home in the bone marrow until they mature. Then they leave and they go into the lymph nodes and the spleen to live there. The T cells are different. T cells are born in the bone marrow, go to their cousin's house to mature, the thymus. And that's why they're called T cells. The T cells develop in the bone marrow and move to the thymus, not thyroid, I said thymus. And they live there until they mature. Then they leave and go to the spleen and the lymph node. So in your spleens, in your spleen and lymph nodes, you will have both B cells and T cells, and they will both be mature, but they mature in different areas. The B cell matures in the bone marrow. The T cell matures in the thymus, which also starts with a T. And the fun begins. Please listen to this. I know it's a long day, but please listen to this. The macrophage engulfs a bacterium and processes it, meaning phagosome, phagolysosome, it breaks it down and takes a piece of the antigen, modifies it, making it look more presentable, you see, and sends it to the surface. Attached to what? Attached to MHC2. Okay? We're talking about an antigen-presenting cell. The macrophage, the dendritic cells, and the B cells. They process antigen, they make it more presentable, and send it to the surface of the cell attached to MHC2, saying, T cell, this is the bad guy. And that's what's happening right here. Which T cell? A T cell that has CD4. This is a helper T cell. So the helper T cell, which has CD4, has now identified the antigen, and by doing that, it activates. Remember I told you, T cells must be presented the antigen for them to activate. Once they activate, they start differentiating into a whole array of different T cells. And each one of them does its own thing. One of the T cells it differentiates to is a memory T cell because it wants to remember this event for the future. Then the other one is a T regulatory cell. This guy is going to regulate the immune system's response. See, you're fighting this pathogen. The T regulatory cell will keep checking. Are we done? No, we still have some. Okay, keep going. Okay, how about now? Are we done? No, we still have some. Keep going. Okay, how about now? Yeah, we're done. Then stop doing it. It regulates the response. It keeps checking. It makes sense because you don't want to go overboard with it. Once you're done, you're done. Stop. Th17, inflammation. Causes inflammation to bring more players. And I told you, right, Jocelyn, I said, they bring the kitchen sink. They fight with everything. So you got Th17, you got the other ones phagocytosing, they got the complement system working, you got the antimicrobial proteins being secreted, you got everything. And Th17 enhances inflammation to make sure everybody comes to the scene to fight. You got Th1, stimulates macrophages to phagocytose. And you got your memory cells, and then you have these guys. Th2 says, oh, I can't forget about the B cell. Th2 will activate the B cell to do its thing. It's amazing how that does that. So once the macrophage presents the antigen to the T helper cell, the T helper cell on the MHC2, the T helper cell activates and becomes a whole array of other T cells. It basically multiplies and differentiates. T regulatory cells regulate the immune system. Th17 for inflammation. Th1 for stimulating the macrophage to start phagocytosing. It remembers the event with T memory cells. And then this one, very important one. The Th2 will stimulate the sleeping B cell that happens to recognize the same antigen. I told you there's a T cell that recognizes this. And there's also a B cell that recognizes this. We just happen to find the T cell and the B cell that recognize the same antigen. You're going to activate the B cell. Look what it does. It multiplies. And it becomes a B memory cell to remember the event along with the T memory cell. So you produce B and T memory cells. You also produce T and B regulatory cells to make sure this response is okay. And then this guy. This is a differentiated B cell. It's called a plasma cell. That's the one that's going to be producing antibodies or releasing antibodies into your blood. It's the same antibody as this one. The only difference is this antibody is IgD. It is attached to the surface of your blood cell. Sorry, your B cell, not blood cell, B cell. And it differentiates into a plasma cell. It's the same antibody, but the only difference is this is attached and that one gets secreted. This one will not attach to antibody. It actually starts throwing it into the blood. So the plasma cell is the one that starts releasing the antibody into your blood. And because it's using blood as the delivery vehicle, it's called humoral immunity. Your antibodies is your humoral immunity, the antibodies in your blood. This is different than another type of immunity that we'll be talking about called cell-mediated immunity. This one here, cells are involved, like the phagocytes, for example. The cells that release cytokines, for example. The cytotoxic T cells, what we're about to talk about in the next slide or so. These make up the cell-mediated immunity. See, the third line of defense has two kinds of immunity. Acquired immunity is two. Humoral and cell-mediated. Humoral is the antibodies. Cell-mediated are all the cells that are doing the killing. The phagocytes, the macrophages, the ones that release cytokines. And these guys, the cytotoxic T cells, the ones that look for virally infected cells and cancer cells. These are called cell-mediated immunity. Hang on, not sure why that slide is in the middle. Let's look at the CD8 ones. So the same event, but we're using CD8, the cytotoxic T cells now. The slide before was CD4, T helper cells. These are cytotoxic T cells. In this case, a virally infected cell or any cell can be presenting viruses here. So a virally infected cell on the MHC1 is presenting the viral antigen to the cytotoxic T cell. See, in the previous one, it was MHC2 that was presenting. In this case, it's MHC1 that's presenting. So it's viral. Viral infection, MHC1, that's a viral antigen. That's a cytotoxic T cell, and that's how you activate this guy. That's one of the main ways of activating this guy. Now, can you activate it from release of cytokines? Yeah, you can, but you can also activate it directly this way through presenting antigen. Again, it's going to produce memory CD8 cells to remember the event for the future. Well, okay, that's the future. But what about now? And now you have an activated CD8 cell that's going to go on the hunt for this antigen. So this antigen-presenting cell is presenting the viral antigen to activate the CD8 cell to remember the event for the future, but to deal with the event right now. The activated CD8 T cell, cytotoxic T cell, is going to go looking for any cell, watch this, any host cell that's virally infected. It released interferon to warn the neighbors, took a piece of the virus, put it on MHC1, and it's waiting for this guy to come and kill it. So that's this cell. Your host cell that's been virally infected has already released the interferon to warn the neighbors, presenting the antigen on its surface saying, hey, when you come by, deal with me, please. I can't hang around for long because I'm not good. I'm no good anymore. So when the activated CD8 cell comes by and realizes, oh, this cell is virally infected, it's going to interact, it attaches to it. And look what really happens. It's not like this guy says, hey, kill yourself like I've been doing here. It actually releases two proteins. A perforin that puts pores in the cell membrane, and granzymes that go through the pores to destroy the cell from inside out. So you're actually destroying the cell, your host cell, from inside out. Don't worry about it, it's no good anymore, it's virally infected. So the CD8 cytotoxic T cell, this is cell-mediated immunity because a cell is involved in your protection here. The porphyrins will put a pore or a hole in the membrane that the granzymes can gain access to your cytoplasm through and cause the cell to die from inside out. What the granzymes do is that they activate apoptosis. And I don't know if you heard that word before, it's called programmed cell death. And that's why I said, hey, kill yourself. But it's not that easy. You actually have to stimulate their apoptotic machinery. So it stimulates apoptosis in these cells. And of course, they die. You don't need them anyway, so they're virally infected. Okay. Here's another interesting thing. You gotta listen to this one. Not like you weren't listening to the other ones, right? Now this one is mind-blowing. I love this part. Because it finally made sense to me. Because we always hear that children are vulnerable the first couple of years of their life. Am I right? We have parents that, you come near their child, you better be like head to toe with antisepsis, right? You better be wearing a mask. There are some that tell you, don't touch my child. Because they don't want him sick, and it's understandable. And you technically have to ask, can I carry your child and give him some bacteria, So, just kidding. Because of the fact that the children their immune system is not fully developed yet. Well, what does that mean? That means they haven't done this yet. Listen to me very carefully. You are born with all the possible shuffles. Do you remember me saying that? So you have a lot of B cells and a lot of T cells that can recognize a lot of things. The red caps, the blue caps, the green caps, the black caps, the yellow cap, whatever. We're always thinking about, okay, these are bad guys. Well, what if they recognize you? Remember, it's just a random shuffle. How will the B cell know that this shuffle matches the red cap antigen and this shuffle matches the yellow cap antigen? No, it doesn't do that. It just creates a shuffle. And if it happens to match the red one, good. If it happens to match the blue one, good. But what if it matches you, the host? That's not good. So now you are born with all the possible shuffles that can recognize antigen, but can also recognize you. Watch this. You can't have these hanging around. In the first 2 years of your life or so, you are learning a lesson in your immune system is. You're learning what is called immune tolerance. You are testing all of your shuffles. Okay? Watch this. I'm going to have everybody that I ask say yes, except for Jeff. Jeff is going to say no. Sounds good? Do you recognize antigen?
Yes.
Do you recognize antigen? You guys get to live. Do you recognize antigen?
Yes.
Do you recognize antigen? You die. See? Yeah.
It's always Jeff.
It's always Jeff, right?
It's always Jeff.
Yeah. It's always him, right? So the whole idea is this. If you're a B cell or a T cell and you recognize host, Jeff, I don't want you around. You're going to hurt me, the host. So I will double check all my B cells and my T cells to make sure that 2 years later, who's still alive? Everybody except Jeff, you see? Because he's a bad guy. He's killing host cells. And I can't have that, you see? And that's called immune tolerance. When you get rid of all your B cells and the T cells that by chance receive the shuffle that recognize host cells, I can't have you around. Sorry. So now fast forward 2 years later, what does the child have? Only B cells and T cells that recognize antigen. Good. He or she just became immune tolerant. We're not done yet. I told you, you have all the B cells and the T cells that you can ever imagine. But only when they come in contact with the antigen do they get stimulated. well, this one did not come in contact, this one did not come in contact. The antigen happens to look like this. And who's got the best fit? Who's got the best fit? It goes with the fit and the color. This guy. See, based on this is the guy that matches this one, not this one. That's called clonal selection. Which one between the clones fit perfectly with this one? This one. So we have achieved the next level, clonal selection. That can happen at any time. As soon as you're exposed to the antigen, it goes through this process. Let's say Haley recognizes the blue one. So now Haley is going to say yes, and everyone is going to say no. Do you recognize this one?
No. No.
Yes.
Okay, take it. I activated you. So what did I do? I did what's called clonal selection. You're my B clones. And I selected you. Because she has the B cell receptor with the variable region that recognizes the blue one. So let's say Tanika recognizes the pink one. And I will look, Tanika, here's Tanika, pink one. And then yellow one, and then green one, whatever. So you're my B clones. You're all of these. The T cell will say, oh, it's Haley's turn to do this. Expand. Once I give you the antigen Haley, you're just one Haley against a million of these. Are you kidding me? You're probably like, Dr. Saad, what are you expecting out of me? You expecting me to go kill a million of these? I'm only one. And well, copy yourself. Multiply. That's this. Haley says, wait a minute. I will multiply. That's called clonal expansion. So after clonal selection becomes clonal expansion, because she realizes I better make more of me, because there's a lot of antigen out there. So it goes through multiple layers here. Watch. First 2 years of your life, you're doing immune tolerance. Immune tolerance, meaning you're getting rid of the cells that recognize self and keeping the ones that recognize antigen. You now have the repertoire of all the B cells that only recognize antigen. Now it's a matter of exposure to the antigen. Let's say at the age of 10, we got exposed to this one, Haley got activated, and she realized I'm only one against a million, I'm gonna expand by multiplying. That's called clonal expansion. And that's what happens there. So once she expands, once she's activated and she multiplies, she will make some of her cells regulatory cells, some of her cells memory cells, and some of her cells will become plasma cells to secrete the antibody in the blood. Now, the one thing I tell you guys, I'm about to say it now, is the antibodies don't kill anything. They don't kill anything. They mark things for destruction. So when she starts secreting antibodies, first of all, she can't be a B cell anymore. When she secretes an antibody, you gotta remember this, she's no longer a B cell. What is she now? She differentiates into what to release the antibody? Plasma cell. Catch that part, please. When she's secreting the antibody into your blood, she's no longer a B cell. She's actually a plasma cell. It's a differentiated B cell. It's actually called a plasma cell. Now some books call it plasma B cell, but that's okay. It's a differentiated B cell called a plasma cell. Remember that the antibodies technically do not kill anything. They typically actually mark things for destruction. So we talked about this, believe it or not. Watch this. The antibodies can bind to the surface of bacteria and at the very least interfere with their everyday life. They slow down their binary fission, they just get in their way. So they may at least make them weaker. But do you really want to kill them? Kill them? Yeah, then do this. Opsonization. So antibodies help in enhanced phagocytosis. They help enhance phagocytosis. How? Remember I told you that the phagocyte wants to kill the bacteria. It finds it maybe a little slippery and it says, I wish I can have, it has like handles on it. Well, now it does. The antibodies that coat the bacteria, they behave like handles so that the phagocyte can grab onto the bacteria better. That's enhanced phagocytosis. That's opsonization. So antibodies coating the bacterium is the reason why we have enhanced phagocytosis, opsonization. Look what else antibodies do. One variable region attaches to the same antigen on this bacterium that is the same antigen on this one. So without this antibody, this bacterium can go this way and that one can go that way. But once you have an antibody linking the two together, they're not easy to move around anymore. You know, in war, they always tell you, contain your enemy. Goal number one to winning a war, contain your enemy. And that's what this guy is doing. It doesn't want this bacteria to be here, and that one here, and that one here. They're gonna be harder to kill when they're all over the place. Do you mean to tell me I have to fight here, and there? No, I can't do that. Contain the enemy and go on the attack. And that's what this guy is doing. It's called agglutination. You're agglutinating the bacteria, making them stick to each other, and that's called enemy containment. And then go kill. How are you gonna kill? Complement system would be fine, or phagocytosis. This is just to contain them. But then you're gonna kill them with all the tricks up your sleeve. One of which is complement system, because it's antibody tagged, so C1 will bind to the antibody and initiate the complement cascade, forming the membrane attack complex and causing the cell to lyse. It's amazing how this works. While another phagocyte on the other end is phagocytosing this, you see? So you got the phagocyte phagocytosing from this side, the complement system bursting these guys open. You're coming at them in all different ways, but at least you've contained them first, contain the enemy, and then attack in all the different ways. Okay, what if it's viral? Neutralize them. The antibodies neutralize viruses by, look at it, attaching to the spike proteins. They prevent them from ringing the doorbell, adsorption. And then eventually your body washes them out. So the antibody neutralizes the enemy, in this case the virus, by blocking the spike proteins, preventing them from absorption. They kind of act like fusion inhibitors. They're acting like fusion inhibitors. Okay, what about bacteria that produce exotoxins? Well, these are proteins, and I can produce an antibody against the protein and precipitate it out. Get it out of solution. Once I get this toxin out of solution, it is no longer effective. It's called precipitation. So they're both neutralization, but they are neutralizing in different ways and different things. This one is neutralizing the virus by blocking adsorption. This one is neutralizing the toxin by precipitating it out, getting it out of solution. All right, secondary response for primary response. This is what we do for vaccination. Initial exposure to the antigen, latent period. That's when you're sick. You're starting to stimulate your immune system. You are producing IgM first, remember me saying that? Then long term, you're gonna turn into IgG. Initial is IgM. Long term is IgG. This is your overall antibody production, the purple one. This is you being sick, and then you fight, and then you get over it. First response, primary response. Now, what happens in the middle of that is memory cells are being produced. Because you just got sick, yes, you fought it, you got over it, but remember, there are memory T cells and B cells that have been produced. And those memory cells will come in play the next time you see the same antigen. The next time you see the same antigen, look, the latent period, if any, is much smaller. You don't get sick really, it may be a little bit maybe, but not as bad as this one, because you've seen it before. So the idea of a secondary response is that it's quicker and more robust, meaning bigger. And you say, oh, I've seen you before. I'm gonna react to right away, and I'm gonna react big. And that's the reason why we don't get sick the second time you see the same antigen, usually. So we take advantage of that when we do vaccination. We inject you with a vaccine to get the first response. You will not get usually you don't get the symptoms, but you get the response, you get the memory cells. So in case you come in contact with the real thing, you've been primed. That's the whole idea. Secondary response is also called antigenistic response. I just report the words. It is also referred to as the antigenistic response. How do we make vaccines? There are so many different ways. We can take the pathogen, kill it, and inject it in you. These are very highly antigenic because they're the whole bacteria. So there are large proteins with a lot of epitopes. They will really stimulate your immune system. But it's kind of like the temporary stimulation because you wash them out and then you lose the effect. The better ones are the live pathogens that have been attenuated. That means they've been weakened. Whatever the bad part of them is, they've been taken out. The good part of this is that the fact that they're alive, they multiply, and they keep stimulating your immune system over and over and over. So, but the doctor will warn you, I'm about to give you a live, but are you okay with that? You may have to sign a piece of paper saying, yeah, I'm okay with it. Because some people don't like that. So there are advantages and disadvantages of both. Here's a disadvantage of this one. They're alive, they can mutate. Good luck now, you see? But I think the advantages outweigh the disadvantages. Because what's the chance of them mutating to something bad? Very little. But is there a chance? Yes. Does it outdo the benefit of it? No. The benefit of it is much higher than the slight chance of it being disadvantaged. So people still take them, of course. The other approach is to take pieces of them and inject them in you. Obviously, the disadvantage here is that they may not be as antigenic as the whole thing. Or genetically engineer the protein. You put it in the bacteria like you did with GFP, but release the protein and inject it in you. Just the protein part should not hurt you. Or take the toxin and denature it, make it a toxoid, or alter it in a way and make it a toxoid and inject it in you to stimulate your immune system. There are so many different ways of going about it. Here's a new way. Inject a plasmid in you. Not me, not this one. Not this one. This has been used in animals successfully there, but they're trying to use it in humans too. But it literally says we're going to have a bacterial protein on the plasmid, a bacterial protein gene on the plasmid, inject the plasmid in you. The plasmid will do its thing, and then now your host cells are producing the bacterial protein. And your immune system is going to say, what's this? I don't recognize that. And it will start attacking it. I'm not going to volunteer for that one. I'll do it the old-fashioned way. All right, real quick, we're almost there, almost there. Gotta at least hear it from me. Adjuvant. Adjuvant is a chemical that is mixed with the vaccine to help in the immune response, in stimulating the immune response. Watch. If I take the vaccine and inject it in you, just the vaccine, obviously it's gonna disperse because of your circulation and the blood moving, whatever. I inject it in you, it right away leaves that site and disperses, so it dilutes out. For some vaccines, that's okay, but for other ones, no, it's not good because it's as if you didn't take it. So somebody said, if I can only trap it there, let it stay there to keep stimulating your immune system before it gets diluted out, and that's what the adjuvant does. The adjuvant is a mixture, something that they put with the vaccine. So when they inject it, it stays pretty much in the same area to keep it in the concentration that it needs to be to keep to stimulate your immune system. These are all the vaccinations. You are not going to memorize this. This is just for future reference when you go to nursing and so forth. These are the vaccines that infant children are taking. Nowadays, slightly younger adults there. And this is me, the old people vaccines. Okay. But these are all the vaccine programs. Probably never thought you would hear this, right? Last slide. Thank God. Can I get a hallelujah? Oh, that was a weak hallelujah. I know, long day. But herd immunity. Herd immunity is when everybody is vaccinated against something, the next person that walks into this room is automatically vaccinated. That's kind of like, why do they call it herd? Because herd is like animals, a herd. But that's what they call. When the whole group of people in this room, let's say we're all vaccinated against TB, the next person that walks into that room may not be vaccinated against TB, but he or she will be automatically protected through herd immunity. We're the herd. That's a common thing. We always, with vaccination programs, we try to reach that level where everybody's vaccinated. That means the next baby is born is automatically immune without actually injecting them. Because who's he gonna get it from if we're all immunized, you see? And there are some people that go, we're against the immunization, the vaccination, and then they ruin that, of course. Okay, the last thing is natural immunity versus artificial immunity. Listen to it, and it will make absolute sense. Natural means natural, something that you get naturally. Artificial means a doctor is involved, or a nurse. Are we catching the words here? Active means you are making the antibody. Passive means someone else gave you the antibody. Let me do it again. Natural means natural. That means naturally given to you. Artificial means a doctor is involved. Active means you are making the antibody. Passive means someone else made it for you. So now we can put them together. Natural active, that's when you get sick. Every time you get sick, you're going through this. That getting sick is natural, and you're making the antibody. It's natural active. Breastfeeding is natural passive. Mom got sick from something before, produced the antibody, put it in the breast milk. The baby is benefiting from that. The baby did not make the antibody. That's the passive part. Breastfeeding is natural. So that's called natural passive. So again, you have to, I'm going to give you examples on the exam. I'm going to ask you, which one is it? And there's some examples for you on the quiz and all that. Am I making the antibody? Yes, then it's active. Is someone making it for me? Yes, then it's passive. Is it something happened natural? Yeah, then it's natural active or natural passive. Is a doctor involved? Then it's artificial. Vaccination is artificial active. Watch this. The doctor is injecting you with the vaccine, but you are making the antibody if you react to the vaccine. there was a recent scare about Ebola, not Ebola, I mixed E. coli with Ebola. Anyways, Ebola, and a doctor came from that part of the world to America, and he actually survived it. And he started donating his serum to other people that came back with the disease there, and he helped a lot of people survive it. So obviously a doctor is involved, because a doctor will be the one that giving you the serum, but whose antibody is it in the serum? Someone else. So when you're getting serum through an IV injection, both a doctor and a donor of the antibody is involved. So it is artificial passive. Are we catching that one? I finished it with 2.5 minutes to spare. Enjoy the 2.5 minutes. All right. I'm going to send you pictures of the answers to these. How about if you take pictures? How about that? Let's just show you the answers, just in case I forget. Good? Good. A lot of questions to answer. You got, again, don't just look at the answers, try them yourself. Are there multiple choice? Yeah. I will see you next, what, Tuesday? I will warn you, I'll be 10 pounds heavier, all right? But I'll be the same person. See you guys.